UV irradiation, a physical method of control. For the UV irradiation exercise, we will be using a red pigmented Serratia marcescens. We will also be using a bacillus, either Bacillus megaterium, cereus, or subtilis. Bacillus is used due to its ability to form endospores. Serratia does not form endospores. You will be comparing the difference between an endospore-forming bacteria and a non-endospore-forming bacteria. To begin, draw a line down the center of your plate. On one half of the plate, you will do a straight line inoculation of Serratia, and on the other half of your plate, you will have a single line inoculation of Bacillus. You will be inoculating 4 plates. One plate will be a control and will not be exposed to UV light. The other 3 plates will be exposed to UV light for different time lengths. The first for 30 seconds, the second for 1 minute, and the third plate for 5 minutes. UV light is a physical form of control of bacterial growth. It is most often used to control growth in air, water, and on surfaces. UV light causes DNA mutations to occur inside of the cell. An accumulation of mutations due to extended UV exposure will prevent the bacteria from replicating, and growth will not occur. When inoculating your plates, be sure to use aseptic technique. UV irradiation works best for surfaces, air, and water. The mutations that are induced by exposure to UV light are caused by thymine and cytosine dimers. These dimers occur when 2 adjacent pyrimidines in the DNA molecule bond with each other instead of their correct base pairing. This makes the transcription and translation of DNA impossible, so the bacteria will be unable to grow. The longer the exposure to the UV light, the more damage is done, and the more mutations accumulate. Bacillus, as an endospore-forming bacteria, is capable of protecting itself from extended UV light exposure, whereas Serratia, incapable of forming endospores, is not. Shown here is a UV light box. This light box uses a wavelength of 254 nanometers. The most effective germicidal lamps will have a wavelength between 200 and 290 nanometers. This is a diagram depicting how your plates should be inoculated. After inoculation, you will be exposing the plates to the UV light. Half of the plate should be covered with an index card and half of the plate should not. Remember, out of the 4 plates, only 3 are being exposed to UV light. One of the plates will be a control. There will be no exposure to UV light for the control. Put your plates in front of the UV light. Place all 3 plates in the UV lamp box. Remove the lids. Once the lids have been removed, use index cards to cover half of each of the plates. It is helpful to use index cards that are labeled with the time length that each plate will be exposed: 30 seconds, 1 minute, and 5 minutes. After putting your plates and the cards on top of the plates, turn on the UV lamp. Make sure when handling the plates that you are wearing nitrile gloves and UV protective glasses. After 30 seconds, you will remove the plate with the 30-second card on top of it. Using a timer is helpful for this. The same will be done after 1 minute. And finally, after 5 minutes, the last plate should be removed from the UV lamp box. Once all the plates have been removed, make sure the lids are on securely and place them in the incubator until your next lab session. If you are the last group to be using the UV lamp box, you should turn it off. Interpretation of results. Here is the control for UV exposure. This plate was inoculated with both Bacillus and Serratia, but was not exposed to UV light. Now shown is the plate that was exposed to UV light for 30 seconds. Bacillus, the endospore-forming bacteria, is still capable of growth and was protected by its endospores from the UV light. Serratia, on the other hand, is a non-endospore-forming gram-negative rod. This organism had no protection from the UV light and was incapable of growth after 30 seconds of exposure. After 1 minute of exposure, Serratia is still not growing, and Bacillus is beginning to show less growth. After 5 minutes of UV light exposure at a germicidal wavelength, neither Bacillus or Serratia is capable of growth. UV irradiation is an efficient method of microbial control in air, on surfaces, and in water. It is also one of the few physical methods that is effective against endospore-forming bacteria.